Okay, I have been so looking forward to this week because this has kind of become my little niche in snow leopard research, I think. And I'm really excited to talk to you guys today about what it's like to actually use genetic methods to study and examine snow leopard diet. So why does diet matter in the first place? As apex carnivores, snow leopards play a huge role in shaping the ecosystems that they inhabit. And they do this in several ways. So first, they work in these top-down trophic cascades whereby they control prey species, which ultimately controls grassland and vegetation. In many habitats where snow leopards are found, there's also several other large predators. And so they're competing for the same or similar resources, thereby controlling one another's populations. And snow leopards also work to prevent mesopredator release by similarly competing with smaller predators such as fox or Pallas's cats. And so this is ultimately going to keep those smaller predator populations down and prevent the decimation of small mammals or bird species. And diet is also a really excellent environmental indicator. So if we're seeing large shifts in the diet of an animal, that may tell us that there's some sort of large shift going on in the habitat that might be worth investigating. And then lastly, throughout many regions of snow leopard habitat, they're sharing this habitat with humans. And so conflicts in the form of livestock depredation are a common conservation challenge with snow leopards. And so there have been several ways to study carnivore diet and several ways that have been employed for snow leopards specifically. Some of you may have used some of these methods. Historically, the most popular one has been microhistology. And this is where you take the scat sample, you dry it out, you pull all of the hair or the bones from the sample, and then you're comparing it under a microscope to a reference database of hairs or bones of known species origins. This can be pretty time consuming. There can be inter-observer bias between different people doing it. And there can also be phenotypic or physical variation within the same species as well that causes misidentification. Interviews have also been done in the past where researchers go out and ask local residents, what's your livestock depredation like? What are snow leopards eating? And as useful as this is and can be something to contribute to knowledge of snow leopard diet, it does introduce a level of bias in terms of asking people, because you're including human subjects, and you're asking them to recall something that they may not recall correctly as well. With GPS coordinates, this is where, so snow leopards that are already collared, you use their GPS information to identify kill sites, and then you can go to that site and actually see what the snow leopard killed. With data synthesis, this is kind of where all of the data that's been published on snow leopard diet is put into a pot, and then we start to make inferences about regional differences or different trends in diet. And then lastly, and the one that I'm going to talk to you about today is DNA metabarcoding, which is where we take the DNA from the scat and we use a diagnostic gene segment that we amplify, and then we subsequently sequence, and we're going to compare those sequences to a reference database of known sequences to figure out our prey species that our snow leopard ate. And I'm going to start with just like a small intro to DNA metabarcoding, and then we're going to kind of go down the line through the process step by step. And so when we collect a scat sample and we extract the DNA from it, and we're asking the question, what did this snow leopard eat? There are a number of possible answers depending on the different region of snow leopard habitat that you're investigating. And so actually figuring out what the snow leopard ate can be quite the challenge. But what we're going to do is we're going to amplify a gene segment that is conserved, meaning that all of our possible prey species have that particular gene. And that's what we're going to in turn call a barcode, as Byron mentioned earlier in the previous talk. You want to make sure, though, that this barcode is actually divergent enough to be able to tell all of your different prey species apart. And so, for example, once you actually amplify that segment, you're going to sequence it using next generation sequencing, which is just a platform of high throughput sequencing that's going to determine the order of your nucleotides for your amplified segment. And then you're going to match that sequence to what you already know to be a known sequence for the prey item. So a reference sequence that you've probably pulled from somewhere like NCBI or the database that Byron walked us through in the last talk there. And so if you have a perfect match, then you can be able to discern, okay, this snow leopard ate a pika. Whereas if you have an imperfect match, then you can rule out a prey item. So for example, that sequence doesn't match with domestic goat. That snow leopard didn't eat a domestic goat. And in terms of walking through the process, this works just the same as, or starts out just the same as any other sort of non-invasive approach. However, I will say that when the scat is collected, it is important to get a cross section. So if you're solely only looking at snow leopard genetics, if that's your only research question, then a lot of times what you're going to really be after is the outside or the casing of the scat, because that's what's most likely to have snow leopard DNA. as it sloughs off the epithelial cells of the snow leopard, moving down the digestive tract. But if we're looking at prey, then we kind of want what's in the middle as well. So I just like to take a complete cross-section of the scat to make sure that I'm getting everything that I can, and then I'm pulling from the freshest part of the scat symbol. And then I extract the DNA as I normally would. Extractions can vary in terms of application or what you're trying to actually extract your DNA for. In my particular case, the same extraction protocol that I'd been using, that our lab developed with a slight series of modifications, has worked for diet analysis and worked for all of our snow leopard non-invasive genetic stuff as well. So I don't necessarily do anything specific because I'm doing diet. And then you need to ask yourself which genetic marker you're going to use to discern snow leopard diet, or which barcode are you going to use. And there are several commonly used barcodes. Some of the ones in dietary studies include markers like 12S, COX-1, and 16S. And remember, you want to pick 1 to 2 gene segments that will be divergent enough between all of your possible prey species to tell them apart. And so the marker that you ultimately end up choosing is going to be highly dependent on the question that you're asking, what prey species are available in the habitats you're investigating, and your overall study area. And a lot of these gene segments, as well as the primers to amplify them, can be found in previous literature. So a lot of the legwork has already been done. So you can pick different markers that you think will work best for your particular application just by looking at previous literature. So for example, 16S or 12S and COX-1, those are all ones that have been applied in previous studies that you could potentially also use for your own work. And remember, because we're working with degraded DNA, because it's found in scat, you're going to want a shorter gene segment. So about 300 base pairs or less is typically pretty common. Some people prefer even lower than that. And you want to make sure that you have reference sequences available for all of your species of interest, so your possible prey species, at that particular for that particular genetic marker that you choose to use, and that genetic marker is divergent enough between all your different species to ultimately be able to discern them all. And if you can't find a genetic marker that fits your needs, you can design your own. So you can pick your own segment of a gene that would be able to answer your question. So for example, in some of the research that we've been doing in our lab, I'm particularly interested in livestock dependency. So it's really important for my particular research question to be able to tell apart all of my wild versus domestic goat and sheep species. So I have to be able to 100% be able to discern all of my possible caprid species within the areas of snow leopard habitat that I'm investigating. And so there hadn't really been a sequence or segment for a gene that had been designed specific to caprids that could reliably and accurately discern them. And so what I ended up having to do was to pull all these reference sequences for all of my possible caprid species that my snowbirds could be eating. I then aligned those particular sequences, and I looked for these constant differences in base pairs, so these fixed nucleotide differences. And once I was able to see, okay, this is a gene, and here's a segment of a gene that's about 250 base pairs long, and there are all these differences, I then designed primers to amplify that segment on the conserved regions of either end of it. And then through a series of tests was able to show that that was something that would work to discern all of my caprids. And so you can use something like that in conjunction with another well-defined genetic marker like 12S to be able to encapsulate all of your possible prey species and to be able to answer the question that you're seeking to answer. And I keep talking about reference sequences, and Byron kind of alluded to some of this in the previous talk. But your reference sequences are not only going to help you determine what genetic marker you should use, so are those reference sequences for all of your prey available, but it's also going to be the template that you use to compare all of your resulting sequences from your own scat samples too. And I like to build a reference file from the start. Building a reference file ahead of time not only helps you figure out, is this genetic marker going to work for me, but it's going to save you time in the long run when you go through your data analysis. And you can find these particular reference sequences through 2 major camps. So there's the Barcode of Life data system as well as NCBI. And so all you have to do is type in your particular gene that you're interested in and then the species. So for example, in this particular search, I wanted to look at COX-1 or my MTCO1 gene and domestic sheep. So I put in COX-1 and Ovis aries. And I got 520 different possible sequences or records from several different areas of the world. And I'm able to just download all of those reference sequences, look at the unique haplotypes, align them all, and be able to get a nice consensus reference sequence. And then with NCBI, you can do the same thing. And so for this particular example, this is just another facet of the website where you can actually search for something like 16S, and I looked up plateau pica in this particular example, got a reference sequence, you download it, and you just do that down the line for all of your possible prey species. And once you've decided on a marker that you want to use, you're then going to amplify it. Typically, you want to use more than one. Sometimes one will do the trick, but in my particular case, I ended up having to use 2 to encapsulate all of the prey species that I was interested in looking at. So you're going to use PCR to amplify those segments. And then once you have that ampliconic product, once you've done your PCR, you're going to have to do a series of steps involved in library prep. So the library being kind of all of your genetic information that you have in your particular sequencing run that you're going to be doing. And so with your individual samples, in the end, you're going to actually multiplex them together. So they're all going to be pulled together into the same little tube at the end of all of this. And so what you do is you add indexing tails to be able to pull those samples apart later. So these indexing tails, for example, here, S502 and N701 for SCAT1, S502 and N702 for SCAT2. And this kind of creates this unique identifier for each species, for each sample, so that everything can be pulled apart later on. And once you actually start sequencing, your next generation sequencing platform, typically the one that's most popular is Illumina. That's the company that does the bulk of this, that makes these sequencers. And within Illumina, there are different sequencer types, and that kind of depends on throughput, how many samples it can do at a time, that sort of thing. In the Unichka lab, we have a MySeq, which is kind of the middle, kind of the sweet spot sequencer. And the way next generation sequencing works is actually really fascinating. It's this process of bridge amplification. And so there's a link there on this slide that you can reference if you'd like to, but I've also put together kind of a supplemental package of all this information as well that will be available for you guys. And so you just load up the cartridge on the MySeq. It's really not that big of a machine. I'm standing right next to it there. You load up your DNA, you put it in, you fill out a few parameters on the computer there. It's just like a desktop PC. And then you do your run, and it's going to determine the order of nucleotides for each amplified sequence from each of the samples that you've put in there. And once you actually end your sequencing run, you're going to get a bunch of raw data. So our raw data on our MySeq just goes to, an external hard drive. I take the external hard drive from the MySeq, I put it on our supercomputer in the lab, and that's going to then allow me to go through my raw reads or my data that I've gotten from my run. And the first thing you want to do is look for quality. So how well did your run actually work? And this is important because you're making these individual nucleotide base calls during your sequencing run, and you want to make sure that those calls are correct. So you're looking for different things like different quality scores. You may trim some of your sequences, as Byron mentioned earlier, so kind of these more ambiguous different nucleotide calls that may not be solid calls. And then you want to import your sequence reference file and your raw data into a program of your choice. And that program that you decide to work with, actually work with your sequencing data with, is up to you. There are free and paid versions. Sometimes people use a combination of both. I use CLC Genomics Workbench, which is a paid version. It can be cost prohibitive, but that has a really nice GUI interface. It's relatively easy to use. But there are other, Python related programs and things of that sort that you can certainly use that are free. And then you're going to actually work in that program to determine what your snowbirds ate by working with your raw data and your reads. So you're going to set a series of parameters, and then you're going to start to map the sequences for each sample that you got from your sequencing run to that reference file that you already created. So you'll import your reference file, you'll import your raw data, and then you'll map your reads. And what read mapping actually looks like, to give you an example, this is a sample this is a sample from Mongolia, EBJ22. And in this particular case, what I tell it to do is, all right, map all my reads to my reference sequence that I've already built. This is where you really save time in the end by doing your reference file up front. So then you're not having to do each individual consensus sequence into something like NCBI. And so, for example, we have all of that bright green coloring that's indicative of more quality read mapping to our actual reference sequences. And then the number of reads also matters. So in this case, we had 82,097 reads that mapped to my reference sequence for Siberian ibex. My other goat caprid species here, my Korean domestic goat, I'm not getting that same kind of solid read mapping there. I'm just getting kind of spotty read mapping. So that tells me that that's not what that snow leopard ate, and I can be confident that the snow leopard ate a Siberian ibex in this case. And sometimes in a single sample, you'll find more than one diet item. It's not very common, but you might find something like a Siberian ibex and a marmot, for example. So that's also something that you might see where you have this really solid read mapping to more than one prey species. And then you just go down the line and you're going to start to kind of build this database of your individual snow leopards and what they ate for all of your samples. And then you can start to actually link all of the prey items to your previously collected information. So presumably at this point, you've got all of your field information that you have when you collected the scats, or that you got from a field collected the scats. And so you can start compiling all of these data that go and feed back into your research question that you can use to answer it. And so what can we do with dietary information? So now we have this big table with all of this information. What can we do with it? And the most obvious is to determine what snow leopards are eating. That may sound really obvious. But here are some really cool things that come out of that, right? You get to know dietary breadth or plasticity, or how many different species, dietary richness that snow leopards are actually consuming. You get to know the role of small mammals and birds. This is something that has not really been well studied with snow leopards. I like to call them snow leopard snacks. So typically we're seeing snow leopards eat really big species, but occasionally we get this plethora of small mammals and birds. And how does that actually play into what snow leopards are eating, and how important are those species for sustaining snow leopard populations? And then you identify the important prey species. So what are the really key species that we need to protect as prey items if we want to protect snow leopards? So for example, in somewhere like China, that's going to be blue sheep, right? Snow leopards eat a ton of blue sheep in China. That's what the majority of my samples come back as. But when I get samples from Pakistan, I'm getting something like markhor and Siberian ibex. And so those are really important indicators of creating conservation species protection plans. You can get differences in diet through time if you resample the same area over and over again, and that can tell you trends in diet. You can also look at differences in sex and individual diet. So as Byron mentioned before, when you do sex identification and individual identification, you can then link back the dietary information with the individual, or you can sort it by males versus females. And you also get an idea of dependency on livestock. So how often are snow leopards eating livestock? Does that vary by sex? Is there such thing as a problem individual, for example, where it's one snow leopard that's responsible for the bulk of the livestock loss? Does that vary by region, time of year? And you can start to look at all of these other indicator factors. You can also see the presence of prey species. So if you're getting a prey item in the diet, then that shows you or infers that that prey item is present in that area. If you don't get it, it doesn't necessarily mean that that prey item's not there, right? You can miss certainly some of those prey items, but it does give you some sort of presence data. And then it also allows you to define unique prey species. So there may be prey species that are not known to really be in snow leopard diet or haven't been reported before or are uncommon. And so you may go in with this bias with these other studies that you're just not expecting to find a certain species. But then when you actually go through with the genetics, so for example, I found a pig in one of our studies, and I was not expecting that because pig had only been reported one other time in the literature. And so it wasn't even in my reference file. It was a study that was, a couple decades old. And then you can actually couple dietary information with different habitat modeling and do some spatial analysis. And this can tell you variation of diet, by different habitat. And you can also look at the influence of different spatial characteristics on diet. So things like anthropogenic disturbances or elevation and all sorts of other things. And then you can also use dietary information as an input to seeing how snow leopards may adapt to climate change. So you can look at the assessment of diet through time to determine dietary changes that may be linked to certain habitat shifts or prey, local extinction events, or things of that sort that you can relate back to mass changes in the environment. And then you can also look at dietary breadth as a measure of potential adaptability. So how well are snow leopards going to be able to adapt when the bulk of their prey species are moving their ranges or have these local extinction events? Are they going to be able to kind of keep up? Or are they too specialists to be able to do that in a particular area? And how does that play into how we prepare for these kinds of things that may inevitably happen? So pros for the DNA metabarcoding approach is that it's accurate. You don't have inter-observer or recall bias associated with microhistology or interviews, and you get rid of the intraspecies phenotypic variation that commonly causes misidentifications with things like microhistology. It's also reliable and it's timely. You can get through this pretty quickly. Not the first time you do it for sure. I'll get into that in a second. But this is something that you kind of just go down the line and you can get all of your information all at once. So it's not like you're kind of piecemealing one sample at a time. You're just getting your data set, boom, it's there, you go, you figure out all of your prey. And then it also provides genetic information of the predator and the prey species. So you may be able to get information for that particular barcode or that genetic marker that you use that can tell you something about the diversity of your predator and your prey species within the actual species themselves. Cons are that it's scat DNA. So the DNA is going to be degraded, and you're also highly susceptible to contamination. This is a pretty sensitive process, you know, from the start, because it's working with scat. We kind of went through all the ways that scat DNA can get degraded through time. But next generation sequencing in and of itself is a very sensitive process. And so contamination is unfortunately an ever-present threat in that particular regard when you're actually prepping your libraries and actually doing the sequencing. And this is why it requires so much training to do. The first time you do it, it can be extremely time intensive. When you don't know what problems to look for, you're going to get, you know, troubleshooting that can be challenging. Some people do not do the library prep or any, you know, you can send these off to a company, no problem. You can just extract the DNA, send the DNA off, tell the company what segment, of what gene you want to amplify, and they'll do it all for you. But that's going to be more expensive. And that's on top of an already relatively expensive process. So this unfortunately can be something that is cost prohibitive, but remember, you're getting a lot of information. You're getting a lot of bang for your buck when you do this. And then there's some information that you're just not going to be able to get from this approach. So for example, you're not going to know the age or the size of the kill. You're not going to be able to look at a scat and, you know, be able to tell that this came from an adult ibex or, you know, a baby ibex, or, you know, this was a particular species that was this really large old male, or, you know, those aren't things you're going to be able to discern. And you're also not really going to get the number of different prey killed, because snow leopards can feed on a carcass for up to a week or 10 days. So you may have 3 different snow leopards that have Siberian ibex in the scat, but that doesn't mean 3 different Siberian ibex were killed, right? It could have been one that all 3 were feeding off of. You also don't know when the kill occurred or whether it was scavenging versus an active kill. So you may have a scenario where a snow leopard scavenged on something and didn't kill it itself. And so is that actually accurate dietary information? It is, but it's not really telling us necessarily predator-prey dynamics in terms of active kills. And I do want to bring up that in our case, only about 30% of the scats that we collect are actually snow leopard. The remaining 70% can be a myriad of other different species, depending on where we're sampling. You can do this with all of the area carnivores. So if you want to, you can take all of your samples that you have and you can sequence them. You're going to get the predator ID as well as the prey ID when you do that. You don't necessarily have to do the prey ID separate from the predator. You can do one big sequencing run and using the genetic markers, be able to tell predator and prey. And that's going to really give you a bigger kind of idea of what's going on in this system, and actually looking at predator-prey dynamics, how all of these carnivores coexist and when placed together, what are the mechanisms for niche partitioning, as well as potential modes of competition between these species. And so just to briefly go through an overview, and this is something that will be included in that supplemental packet I mentioned earlier. We're going to collect and extract our DNA like we would pretty much for any other application, really focusing on getting that nice cross-section of the scat so we have both our predator and our prey DNA present. And then we're going to amplify our gene segments. We're going to pick which one is going to be best. It may be more than one. We're going to pick that, we're going to amplify it, and then we're going to use next generation sequencing to get resulting sequencing reads of each of our samples. And then we'll analyze all of those using different statistical softwares and online tools, and which you use are kind of up to you. There are different avenues to do that. But the idea is that you're going to be able to do this read mapping process where you can determine, yes, this is a snow leopard, or if you're doing more than just snow leopards, you know, what is the host of the scat and what that actual host ate. And then you're going to go down the line, compile all of your data, and you can do a series of different statistical tests, habitat modeling, and get a really good idea of what's going on in the system. And of course, you're going to share that information with your different stakeholders that are involved in the study, and live with snow leopards and share these landscapes with them. So things I want you to walk away with, DNA metabarcoding can improve our knowledge about snow leopard diet, and it's going to give us accurate information. It's going to give us kind of a more holistic picture. We're also going to be able to provide a lot of information, and this is all information that's really important in making effective and long-lasting species survival plans for snow leopards. And DNA metabarcoding as an approach can be done quickly and reliably, but the upfront kind of learning process can be really intensive, and it's going to require some training. And it is a process that is expensive and will require some upfront costs. But like I said, you get all this really nice information, and you can work with that data set to answer several different questions. So it's really kind of an investment, I like to think of it as, in terms of the studies and the questions that can be answered using this particular technique. And that's all I got. So I don't know if we have time for questions or not, but I'm happy to take any.
Yes, there is one question that came up real quick that I'll just read for you from Luciano. So he asked, Charlotte, is there a way to quantify the amount of prey DNA and get information on Relative composition of diet, so if relative abundance actually reflects the frequency of that item in the diet, or does the amount of prey DNA reflect the recent meal? So to get at a bit of those questions.
Yeah, so there are some people that will say that the abundance of reads that you get that map to a specific species are indicative of how much that, or how the abundance of that prey in the habitat, or the amount of prey, the number of prey that you find, are indicative of what you would find in the environment. I'm not so sure about that. I do think, for example, that there's a lot of, sometimes it's really obvious, right? We get a really high occurrence of blue sheep, and then yes, that's going to be, the species that we expect to find in the landscape. But I'm not so sure that relative abundance actually reflects the frequency of that item in the diet, or if relative abundance in diet reflects what's actually available in those habitats. I'm also hesitant to say that the prey DNA reflects a recent meal. I guess if you get a really high quality scat sample and you know it's fresh, yes, you can say that kill was made relatively recently. But we're still, I feel like this is the big question that I would love to answer within like my lifetime is, how do we age scat? How do we reliably age scat? Because it would really answer so many questions. But if it's a fresh scat, maybe, yeah, within the last couple days or week, or I watched a Tibetan fox defecate and then picked up that sample. So yeah, that would tell me that was a recent kill probably, but those are very rare events. And when reporting data, I count how many times a given item is found without accounting for DNA quantity. Yeah, so I just do frequency of occurrence, if that answers your question. I'm not sure with the DNA quantity part what that exactly is alluding to, but I just do frequency of occurrence.